Thank you, everyone, and welcome on behalf of Dr. Purvish Parikh sir and myself to one more chapter of OBTO, that is Oncology Beyond the Obvious. Every time we say this, and every time we get flattered by the response of the people when they, before and after the OBTO, they tell us. Today also one of our colleague just messaged me in the afternoon that thank you Amish, you make us read more and you make us listen to new things, and that is what our endeavor always is. Dr. Parikh sir and myself, before every OBTO chapter, we try to see what is new today and what is it that we need to learn from each other. And that's when we select the particular topic. As always, the pattern of OBTO is first we have an interview, then we have a panel, and after that we have an expert opinion. Today, Parikh sir and myself, we have been discussing about this for last, I think, 6 months now. And today, for the first time in OPTO, we are trying to implement this, that there are two interviewees, and both have their own expertise. And what sir and myself, we said that let us try to extract maximum out of these expertise, and let's see how much we learn out of this. So today we are discussing a very, very important topic, because NGS has become a flavor of oncology for last 2 years. There is a plethora of labs which have sprung up. Every day one new lab comes and meets us and says that, sir, we do NGS on this platform and these many genes and RNAs and DNA. We really feel that how much it is applicable in a day-to-day clinical practice. So today we are going to discuss about molecular targets and FDA-approved genome-targeted cancer treatments. Uh, Interviewer is Dr. Purvish Parikh sir. I think, I always say this, anybody who needs an introduction, who's Dr. Parikh, probably is not an oncologist either by degree or by experience. And I have always had the pleasure of introducing sir. But today, since he's an interviewer, I would give a very brief introduction. He's the backbone or the spine of OBTO. Without him, I don't think this program can go forward even for 2 months also. So, sir, thank you so much for being the co-program director also and guiding all of us to the best of your capacity. Uh, and I would give the platform to you, sir, to take this forward and introduce our interviewees and go forward, sir.
Okay, thank you. I'll start by sharing my screen. Can I share my screen?
Yes.
Okay. So I hope that the screen is visible.
Yes, sir.
Okay. So thank you very much, Amish, for that kind introduction. As Amish mentioned, today we have two interviews. So first we have Dr. Kumar Prabhash and then Dr. Chirantan Bose. And the reason why we selected this way is because today's topic is such. It talks about molecular targets and then targeted cancer therapies. So we definitely needed expertise on both sides. And it's a proud privilege to have these two people. And Dr. Kumar Prabhash has been my colleague in Tata Hospital and has risen to great heights. He is authority on several aspects, and particularly so for doing clinical trials that are critical or crucial in the Indian context. Just a few days ago, he was talking about borderline head and neck cancer patients and how his study has revolutionized how the world treats them. So let me start with Kumar Prabhash first. So before we start with the interview, as is usual, I will share some key highlights from the publication that we are talking about. So the flowchart of the search selection is that they looked at 383 indications in across 8 years. 238 were eligible indications. And out of these, 206 could be included in this study. And these 206 indications were across 84 genome targets. Now, what was the strength of evidence? The strength of evidence was according to the European Society of Medical Oncology scale for clinical actionability of molecular targets, that is ASCAT, and ESMO's magnitude of clinical benefit scale. In this scale, substantial clinical benefit was grade A or B for curative intent and grade 4 or 5 for non-curative intent. And the molecular targets that qualified for ASCAT category level 1A or 1B were thought to be associated with substantial clinical benefit by the ESMO MCBS and were rated as high benefit genomic based cancer treatments. And Aju has written several articles about this. Now, these are the 84 with FDA data categories. This included 67 new drug applications and 17 biological license applications. The approval was either initial in 37 or supplemental in 47. And the regulatory pathway was standard in 52, breakthrough therapy in 50, orphan drug 58, and accelerated approval 32. And you can clearly see that there was a lot of overlap. But very interesting is that some of the standard regulatory pathways was on single arm study. That was a significant number of 25%. And lo and behold, single trial supporting approval was in as many as 86%. Now, 50 molecular targeted drugs for 84 indications. The indications were approved based on phase 1 or phase 2 pivotal trials in 54%, single arm pivotal trials in 54%, and subgroup analysis in 57%. The primary endpoint was overall response rate in just about half and median duration of response in 57%. So the regulatory approvals was primarily based on surrogate markers, that is disease-free survival, progression-free survival, and durable response rate. And they also included basket trial design where clear approval was received. Now, this is the ASCAT class distribution. On the left-hand side, you can see ASCAT class for trials, and on the right-hand side, you can see ASCAT trials for data points. Now, on the horizontal axis, that is 1A, 1B, 1C, and 2A. So, 1A and 1B are important. They represent prospective randomized trials and prospective non-randomized trials. 1C was for basket trials, that is tissue agnostic trials, and 2A was for retrospective trials. Is clinical benefit substantial? That is the moot point for this publication. And 38 out of 84 trials had 1A ASCAT targetability and 32 had 1B targetability. Overall, 29% trials demonstrated substantial clinical benefit via ESMO MCBS. So, 29% were, in conclusion, were associated with a high benefit genome-based cancer treatment. So now let me start with Dr. Kumar Prabhash and ask him the questions. So rapid fire, do you agree with the methodology employed in this publication?
Hi, thank you, sir. Thank you for the opportunity. So first, do you agree with the methodology employed? Sure, any methodology is useful, but as far as the interpretation is concerned, methodology was not the best one.
Right.
Do you want to elaborate, or you can ask me later? I'll leave it to you, sir.
Yeah, we'll go through the questions and then we can elaborate later.
Yes.
So the second is, are ESMO MCBS and ASCO value framework interchangeable or not?
So looks like as far as understanding the benefit of patient is concerned for the therapy, there are different results. There are many places looks like they correlate well, but there are a good number of papers suggesting they do not correlate well as far as the understanding of benefit is concerned.
All right. And obviously, there are some differences. Like ASCO, VF takes into consideration the cost of the drug for 1 month, whereas ESMO does not look at that. So the third is companion diagnostic test sacrosanct? That drugs cannot be used without it.
Sorry, definitely not. We will be happy to discuss it once we have a discussion.
Okay, okay. And should FDA change the label once confirmatory trials show a better or a poorer outcome?
Yes, there is a merit to it after a thoughtful process. What kind of expectation was there from the trial and by the FDA, and that may decide what happens to the confirmatory results.
Okay. And the last question is that tomorrow morning when you go to Tata Hospital, will you recommend some changes to your colleagues based on this publication?
Definitely not.
Okay, okay, perfect. So now we have gone through the rapid fire, and then I will request you to elaborate on any of these points that you think are appropriate.
So, coming to the first one, do we agree with the methodology employed? The moment we think about substantial benefit, now the best benefit for what we have, the single arm studies, is grade 3. Now, the challenge is in today's time, what we have is you have a small group of patients, rare tumor, where the benefit is substantial. And we have, for example, craniopharyngioma. If you give a tablet, you have 100% survival. Now, again, they will belong in grade 3. So there is an issue that the moment you have some index being developed, they also need to change with the time. They did change. The first MCBS was different from the second one, but looks like there is a need for change.
Right.
Is ESMO and ASCO interchangeable?
Go ahead.
Okay. So ESMO and ASCO, sir, you commented about companion diagnostic. I do understand the companion diagnostic. We want to have best of the tests available, but best example is for them is NGS testing, or for that matter, EGFR testing. We didn't have a companion diagnostic. Otherwise, we will be bound by the test and by the company who markets it. Otherwise, individual lab have a chance to develop it, and we need to maintain a quality. But then we will have a cost-effective test being available. Otherwise, the test will go through the roof. Should FDA change the label? Sorry.
Go ahead, go ahead.
Should FDA change the label once confirmatory trial shows better or poorer outcome? Now, poorer and better outcome has a whole lot of context. For example, if 100% patient crossed over and then you have a different result, that is not should be the basis for changing. But interpreting the trial and changing will be required in many places.
Okay. So since this had majority of instances was surrogate markers and did not look at either overall survival or quality of life in the majority of instances, should we also allow this for conventional chemotherapy or immuno-oncology drugs? What do you think?
Sir, I'll give two examples, and that may make us understand. One is our work, which was triple metronomic chemotherapy. When it was first presented in ASCO, it was a phase 1/phase 2 trial. We had not published it. Up to date had picked it up, and it was one of the options in the up to date guideline of that time. The reason being that at that time in platinum refractory disease, having a response rate of 40% was not there. There was no documentation of any other combination or treatment which had this kind of benefit. And that was a chemotherapy drug combination. So yes, if your chemotherapy drug also provides substantial benefit, or it can be any treatment which provides a substantial benefit as compared to existing benefit treatment, then yes, they should also be considered with a similar approach.
All right. So when your options are in the limitations, as far as the trial or other trials is concerned, garbage in and is same as garbage out. So there is somebody who has said that once the drug receives regulatory approval, it automatically achieves tier 1 rating in ASCAT scale. So is this like a self-serving manner in which people are getting their drugs approved for use in clinical practice?
There is a merit in this statement, and I'll elaborate a bit. So we can
Yes.
have scales. Think about, we had a scales, one side of the Atlantic being developed differently as compared to other. Why couldn't
Yes.
they agree upon one scale? So that tells you that they do have differences of opinion and they wanted to develop separately. It's a different matter. We don't have a scale of our own, but a whole lot of common sense is there, which prevails, and that's the reason.
Right.
In spite of most of the trials not reaching substantial benefit, they have approval across the Atlantic from the regulator to be used, and that's the reason majority of the clinician uses this in spite of this being there for now almost around a decade, these scales being available.
Understood. Understood. So, ESMO MCBS has developed guidelines when they have to evaluate single arm studies. And for single arm studies to receive the highest efficacy score, they have recommended median progression free survival of more than 6 months, or overall response rate of more than 60%, or a combination of overall response rate between 20 and 60%, plus a duration of response of more than 9 months. And if there is any safety concern, they will downgrade the score. And if any additional data is available for quality of life or confirmatory phase 4 data, then they will upgrade the score. What do you think about this for single arm study?
So what it tells us that in single arm study, by and large, whatever you may do, you will remain at grade 3. And
Right.
this is one of the limitation of this whole analysis, because almost 60%, if my number is not wrong, if I remember correctly, where the treatment which was approved based on single arm study. And that is why
Absolutely.
in the whole analysis, this doesn't look exciting for as far as the approval is concerned by regulatory, because these were the majority of the drugs which is there. And yes, we need to remember, now single arm studies was not included in MCBS when it came initially. Later on, they did realize that this needs to be included because there are now drugs being developed which give substantial benefit of response rate of 60%, 70% in a second line, third line, and beyond treatment. So yes, I think as the time goes by, hopefully the same group will realize that we need to upgrade this whenever you have a response rate of 60% and 70%, not classifying them as a substantial benefit may not be the best way to put the benefit for them.
Very good. Very good. So, your opinion on using these conclusions, that government authorities, payers, insurance companies use this method to allow or prioritize availability and reimbursement of only those molecular targeted therapies that have high value. What do you think?
So, my opinion is that on both sides of Atlantic, the authorities, the payers, the insurance companies, they have maintained the sensibility to take care, to notice this, but they are still waiting to act upon it. So, sure, the people who developed it in those own area, they have been wary of implementing in the clinic and implementing as far as the as far as the rules, or you can say policy of the government insurance is concerned. So that gives us a clue that what we should do.
Absolutely. So this is not something that is practice changing, which you have already mentioned. Now, there is evaluation of changes of the benefit over time following reimbursement use. And you have already covered this, so we'll skip that. And you can see here that the overall survival available less than 12 months, more than 12 months, and more than 24 months has been used in this publication. But even more important is this article. And I think, Kumar, you are the right person to guide us on this. There was your previous colleague Vijay Patel and has published this on cost effectiveness of temozolomide for treatment of glioblastoma multiforme in India. And this was in JCO Global Oncology. And here the conclusion was that the cost of temozolomide in India should be reduced by 90% before it can be used when it is used with radiotherapy in a concomitant fashion, followed by maintenance temozolomide. What do you think about this?
So, yes, first of all, disclaimer, a very dear friend, Dr. Vijay, and we had extensive discussion, and that's the reason I could remember it, this whole publication. And he was very keen on knowing what he should do and say, hey, better be part of it. And this is a collective decision, so one should go ahead with that. But I can tell you what I'm saying was the same opinion by him. So the cost at that time of temozolomide was something few thousand, something around 2,000 rupees, something around per month. And the interpretation of, and this is again based on some metrics which has been developed. And when the metrics is applied, what is expected that the cost should go down by 90%. What does it mean? The cost of the treatment should be around 150 to 200 rupees per month. Now the challenge is, will drug will be available in the market ever like that? Now, that we should remember about it. And then we also checked it, and I had asked him, and then I asked few of my colleagues around, is there anyone in your hospital, at least in our hospital, who is not able to take the treatment? And he had told Kumar, everyone is able to take the treatment. Suggesting that metrics we may develop, but we should not be blind to it. Best example is sunitinib. When NHS and NICE had evaluated sunitinib, for its efficacy and for able to market for the patients and available as a part of NHS in UK, it is a very nice summary. Anyone can read it. How they had talked to the clinicians, the patients group, the social worker, and taking all those in consideration, they had changed the quality for sunitinib, understanding that it was useful for patients. Again, highlighting that you can have a metrics, but ultimately metrics is servant for the what the purpose it is for, and here it is patient care. If the patient care is getting compromised, then metrics should be changed, not the patient care.
Understood. The other article that you already mentioned is this, that BRAF MEK inhibition in newly diagnosed papillary craniopharyngiomas. And that is because this activating mutation is seen in almost all the patients. And this was a single arm phase 2 study. But lo and behold, almost all the patients had responded. And the only person who did not respond was the one who took the treatment only for 8 days. And if you look at this, this is the event-free survival and this is the overall survival, which is 100%. So what is your opinion about this, Kumar?
Sir, this is a classical example of uncommon tumor where you don't have a treatment. There is no other way of developing treatment. And if you have these kind of data, idea should be do whatever, see that how the patients can receive this treatment. If you want, sir, because I treat another group of patients, we call amyloblastoma. Again, BRAF mutation, again the similar drug, 80% patient has BRAF mutation, 80% patient responds. And based on, again, a small series, it is a standard of care. Reason why, if you don't give it, those patients loses half the mandible. And losing half the mandible, one needs to see patient, follow them to realize what happens to them. So again, a good example of how we should be changing the metrics to see that our patients benefit.
Perfect. Thank you very much, Kumar. I will now move on to the next interview, Dr. Chirantan Bose. As all of you know, he is a molecular and precision oncologist. He was working in the industry first in the diagnostic lab, and now he is very much part of the MOC group. And Chirantan, I will again start with you with the rapid fire. These are the 5 questions. I would like you to briefly say yes, no, and then we'll move forward to your discussion.
Thank you, sir. So I concur with Kumar sir actually on almost all of them, with slight difference of opinion in the 5th point. So methodology, I think every methodology has its pros and cons. There was certain intrinsic biasing in the methodology of the SCAT MCBS. The way the method was designed. So I have some reservations on that. I think clearly, if you ask objectively, yes or no, between MCBS and ASCO, they are not interchangeable. There are some very significant differences in between them, so they are not interchangeable. Companion diagnostic obviously is not sacrosanct. One of the examples which I come across very commonly in our practice is, the use of CDK4/6 inhibitors in hormone positive breast cancers now for the maintenance, especially where we are not actually looking at. And of course, across immunotherapies, there are so many examples where we do not use companion diagnostics. So I think everybody would agree that we need more dependence on FDA. So FDA change of label or at least some kind of flagging is definitely, I think it should be a welcome and much wanted outcome. I feel that, at least where generalized molecular excavation of data and correlation with available therapeutic options is being practiced, I mean, at least in our center, we see that it is impacting practice. Maybe not in a very significant way, not a Monday morning change, but it is definitely in its small way impacting.
All right. So should I say, and I'm asking whether this statement is correct or wrong, that any patient who fulfills the criteria shown in this publication, we are reassured that he will get significant clinical benefit. And if somebody else whom in our wisdom we feel that they might benefit, but he is not included as part of this trial as showing clinical benefit, we can still go ahead and try in a particular case based on the case's specific features. Do you think that's a fair statement?
Yes, sir. Actually, one of the major unanswered questions in this, which you have also highlighted, is that it was all single arm, data, which we consider is biased. So does a KRAS G12D mutated lung cancer patient or pancreatic cancer patient does better with chemotherapy or not is the question which this has not answered. So I think, yes, that clinical opinion rests very strongly.
Now, you're the best person to answer this question. How useful is the SCAT ranking system for stratification of molecular findings with respect to therapeutic recommendation? And if you find it useful, do you add this to your real world report?
Sir, actually, again, I totally concur with what Kumar sir said, because we know that there are differences in different parts of the world. If we come to that, when we sit down and we want to make Indian or Asian specific guidelines, we do see that we have various different points also which we want to include or not. So I wouldn't say on a perfect scale, but it is always good to have some benchmarking. Say, for example, what do we do with the 50,000 cancer patients who, are treated at our center? So some benchmarking is important. Now, whether we follow ASCO or whether we follow SCAT is this thing. What SCAT has done actually is they have created various interactive platforms which ASCO does not have. So it makes a little bit of interrogation more interesting for us on day-to-day practice. So I mean, we are still exploring.
Okay, right. Now the Rome trial is something that is quite different from what was published or what is the trial that we are talking about. So would you like to walk us through what is your perception about the Rome trial?
Yes, sir. So this also was a significantly Europe-centric, study, of course, which had involvement from US as well as Canada. The final findings of the Rome trial were presented at ESMO a few weeks back. The penultimate data was presented at ASCO. Contrary to what the SCAT criteria has been talking of, this was actually a double arm study. So there was a control arm all through. And actually on what I really liked is that if you see at the right end of the algorithm, there is a crossover also allowed. So it was very patient centric in the sense that it showed really that ethos that they are working towards welfare of the patients. And so that is why that initial thought that this is pharma driven, that went away. All these are fairly limited in a way. This Rome trial actually involved about 22 genomic markers, and which were for target drugs, and 2 markers for immunotherapy. And there were about 23 target drugs and 3 immunotherapy drugs overall, which were matched. So this is a classical basket trial, which then was extensively discussed through Molecular Tumor Board. And finally, there was a decision arrived. So out of about 1,800 screened individuals, finally, I think 400 individuals were randomized, which were divided into 2 arms of 200 each, 200 who received standard of care treatment and 200 who received target treatment. And this is the number of mutations against the target drugs, which I was talking about. So overall, it looked a fairly balanced study based on what is currently available in the market. So of course, there were the outcome was fairly strong for immunotherapy, and TMB came out as one of the very strong indicators from this study. So my thought overall is that, but it is interesting for everyone to read through this Rome trial, because it in an unbiased way shows the implication of comprehensive NGS and potential options, at least, that come out.
Perfect. Thank you. So Kumar and Chirantan, both of you, thank you very much. I did not have too much time to ask in-depth questions, and there could be many more comments that you would have wanted to make. Unfortunately, we are already overshot my part of the program. So I will stop my screen sharing and I will hand over to our moderator, Dr. T. Raja. Again, he is somebody who needs no introduction. He has been with Apollo Chennai right from the very beginning, and he has, developed the program, taken it to great heights, helped develop the Proton Center, and he is an excellent moderator. When we always want to have him with all our scientific activities, so that we can benefit from whatever he is bringing to us. So I will hand over to Dr. Raja with for the next part of the program.
Sir, thank you, sir, and allow me to share my slides. I think my slides are visible and I'm audible.
Yes, we can see your slides.
Yes, sir. As usual, sir, Purvish, sir, and Amish, I mean, I am always so fascinated by how you ever thought about this OB2O and how you are sustaining. Every OB2O is such a good eye-opener, learning, and thank you so much on behalf of the broad community, and excellent panelist you have given to me. So I will really delve right into, but you have covered many of my questions with the 2 elite interviewees. There will be a bit of an overlap, but that should be okay. So we want to discuss the topic broadly about the JAMA paper, but also about genomics, whether in general it's good, bad, or ugly, either use. That's how I'm going to take. So today's discussion, we'll try to cover these aspects. We'll try to cover the, quickly about, our take on FDA approval processes. And then we will try to talk about the SCAT and the ESMO MCPS, as Purvish sir already covered. Then we'll go into the JAMA paper, methodology, and what do we get out of that. Then we'll briefly touch upon the author's contribution to this science. And then our collective clinical experience, what does it say? I mean, how do we interpret this paper and these kind of papers on our clinical experience? And how can we explain some of the discrepancies, and what do we do in future? That's how I want to take this discussion forward. And therefore, my first question is based on the FDA approval process. It's quite time consuming. Everybody knows the drug, once it starts to make the journey, it takes quite a bit of time to finally get the approval, and it goes through multiple obstacles before it reaches. So in general, it is known that 90% of the trials of the drugs fail clinical trials, and as you go on from preclinical to so on, so there is a very steep curve rise, and many failures are happening. So finally, who reaches the podium is already very small, even though the community and oncology in general needs new drugs all the time. So, but there is a real challenge about how do we try to overcome this particular obstacle. My first question goes to Dr. Sahu. We are in need of drugs, but finally it has to cross all these bars and come out. Many drugs fail clinical trials, and so how do you look at that, Dr. Sahu?
Sir, I feel it is necessary because ultimately these are meant for patients in practice, and the scrutiny that the patient goes through in a trial and what we do in a clinical practice is very different, sir. So trial does collect a lot of the data sets, and most of the trials now are looking at even patient quality of life, a data set which we normally don't look at in clinical practice as stringently as it is done. So I feel uh, the trial process has been there for quite some time, and it has passed many decades of testing. And I think the scrutiny is required because once it comes into a pre-market, then the drug is available for everyone, and sometimes it can be of no benefit, and toxicities can be there severely. And we have seen even after this scrutiny, drugs approval has been withdrawn. Bevacizumab in breast cancer is one of the gold standard, what we know that. So yes, this is a good thing, should happen. I would I put one step further. I think the way FDA is approving, I hope we discuss that, probably also needs to be scrutinized more further, because the way the rapid approvals, accelerated approvals happening, and once they approve, there's a beautiful data set. If I can take under 10 seconds, there's a publication that happened last year, that once FDA has done a approval, maybe accelerated, based on that we will have a full approval, and the company had withdrawn 11 approvals, and still NCCN guidelines continue to have a category of recommendation even after the approvals are withdrawn to the company. So I think we are doing good. We should be much more stricter, because these drugs are not only costly, are also toxic, and we should have a very good scrutiny, and approval should be rigid. That's my take.
Thank you, Dr. Sahu. Your point is you are happy with the being strict, and you in fact want much more stricter. So I will now look at like what you said, but there is a I'll come to everyone. Chirantan, I'll come to you. So the point is, FDA takes it through a process, and as we know, they try to strike a balance. You can't be too careless. There will be toxic deaths, and then you can't be too cautious. But there is a need to bring the drugs out, and they need to have some endpoints. Therefore, all these endpoints in overall survival, the gold standard, but we all have, they have the ORR, TTP, each one has their benefits. Time, for example, if you just take ORR, then it can be brought about in a short duration of time, and there is a very high certainty of tumor reduction being directly related to the drug. That's the beauty of ORR, but it has its own peril. There are many examples. There is this oxaliplatin example, and there is the example of crizotinib, where the magnitude and what happened in the clinic were discrepant. And with all these things, considering what FDA is able to do and not able to do, usually they use these endpoints, and typically they look at large randomized clinical trials. But though you don't need a comparative efficacy as a requirement for approval, non-inferiority trials are allowed, overall survival is not required to grant approvals, all these things. So I just want to ask Dr. Randeep, is this all okay, how FDA takes it? Broad comment. And is there anything else that you want to specially say, what the FDA does? Dr. Randeep.
It is very difficult to give a broad comment because the response rate and PFS or whatever surrogate endpoint is used is very important that in what context we are using. I mean, if I'm using response rate as a criteria for a tumor like Kumar sir mentioned craniopharyngioma or amyloblastoma or a rare sarcoma, like an EZH2 inhibitor being used in the sarcomas, that is fine. But tomorrow, if I'm using a response rate for a breast cancer or a PAT-CR for a breast cancer for a decade, we have not been able to come. PAT-CR is a refined form of response rate, you will agree, and we have not been able to sort out and not even a randomized or a level 1 evidence to show that PAT-CR is improving survival. Even PFS also, I mean, these are detailed comments, I know, but PFS is also like if I'm using PFS for a very small tumor like a pancreatic cancer, why don't we look at OS when the PFS is 4 months and an OS is 1 year of a second line pancreatic cancer? Why I would need a PFS? But on the contrary, for a hormone receptor positive breast cancer first line, I am okay if they are using PFS as a surrogate endpoint. So surrogacy and endpoint, and I think FDA knows better than us, the experts sitting there know better than us, that in what clinical context they are putting that surrogate. If so, tomorrow they put response rate for any breast cancer, I think the whole community will understand. And their duty is to maintain a balance that particular endpoint of surrogate. Example, PFS only I told you in pancreatic is different from breast. And in ovarian cancer, we have already seen that PARP inhibitors in the later lines of therapy were approved on PFS. And then all three PARP inhibitors were pulled down because of inferior OS. And then you feel that guilt that so many patients have put on PARP inhibitors. And now how do you tell them that you have a hazard ratio of 1.2 and you may die? And so then that is what Sahu was telling, that yes, the process has to be strict because once it spill over to millions and across the globe, then it is very difficult to withdraw back. I mean, for me to tell someone whom I have put on olaparib 2 years prior and now tell them that you have more chances of dying because
Yes,
you
but Randeep, I agree that, there seems to be, on one end, there seems to be, a lot of need to be flexible, but at the same time, we are all very afraid that over flexibility can lead to a lot of. So everyone seems to be agreeing with what is happening so far, but there is some worries about the process. In this context, we are going to look at the papers. That's why I want to, Purvish sir, I'll come to you. So now I'm going to move on quickly into the SCAT. Our Chirantan spoke about this. SCAT is comprehensive, reproducible, and it was published quite a few years ago, 2018, and it is a good framework to rank the genomic alterations as targets, and it's comprehensible, it's reproducible. Let's see what it is. So these are the SCAT pyramids 1, 2, 3, 4, 5, and 6. So my question is that I'm trying to ask Dr. Amit Verma, if you are there. The question was asked to the laboratory providers, but as a clinician, do you routinely look at SCAT, these scores, and would you apply it on your day-to-day clinical practice when you look at this, Dr. Amit?
It's a very interesting question, and no, there's no straightforward answer. The reason being that whenever we see a target where we have a SCAT level 1, we are already doing those things in a clinical practice. So the simpler questions are already been answered in our clinical practice. So practically, we do not require this SCAT scoring that much. But once we go to something more complex, SCAT sometimes is not able to give those answers. So for all practical purposes, it seems very fair to see a target like HER2 and SCAT category 1 that is well understood to all of us. But again, but for other recommendations like PIK3CA or CDKN2AB, those things which we want to apply into our clinics is absent in the SCAT. So
at least pertain to genomic alterations, we'll stick to that at this point.
Yeah.
These are already available in the ESMA websites. These are where, these are easily accessible to us when we look at it. This is for the lung. This is the, SCAT1 recommendation for the genomic alterations for the non-small cell CA lung. And this is for colorectal cancer. And this is for cholangiocarcinoma, gastric cancer, and prostate cancer. I'm just broadly illustrating these things. I'll turn to Dr. Ajju. Dr. Ajju Matthew, Do you routinely recommend look at these in your clinical practice? I asked the same question to Amit, but I want to ask you also. I mean, what is your application when you want to look at these genomic alterations, and then how do you take it to clinical practice, Dr. Raju?
Yeah, thank you for that question. More or less, these SCAT guidelines are quality thresholds. So it does help a clinician in his practice when they see genomic reports of which they may not know the finer points of how a target works and how what drug works. Sort of for a quick a clinician working in a busy setup, this sort of provides them a quick evidence update on what is known about what targets and what depth of activity. But at the same time, there are exceptions. There are definitely exceptions. We all know and we have seen that in practice. So that is where I think molecular tumor boards will help. And sometimes in the setting of a good molecular tumor board discussion, maybe there may have been an indication where an SCAT category 2 drug may be trialed in a patient even outside of a clinical trial. For example, ATM here, as you see, it is 2A, which is not strong. But at the same time, there are some PARP inhibitors that may work for, in an exceptional way, in meaningful manner for a patient. So it does give the clinician a quick perspective into the level of evidence that exists for a certain target and a certain drug.
Thank you. I know Sahu has got a point. I'm going to come to it in a minute, Sahu, to view. There are some more publications and guidelines if you want to access and look at what is the SCAT recommendation for individual malignancies. SCAT 1, 2, 3, these are available. These are for the lung, and this is for the breast. And if you want everything, endometrial, all these are available. Fine. Now we come to the crux of the matter. How applicable are SCAT in day-to-day practice, as Dr. Raju was telling, and into molecular tumor board? So I'll come to Sahu. You wanted to raise a point, and but
Yes, sir. Can you have that slide of prostate
SCAT,
cancer? Can you have the slide of the prostate?
SCAT,
Sir, can you have the slide for prostate cancer which you just showed to Ajit?
Yeah.
So if the short answer is, I normally do not look at SCAT as such before we do in the practice. Yeah, here. If you look at it, it broadly tells you that if you're looking into this, maybe the 1A and 1B is something that is of clinical relevance. Even 1C and 2A becomes down the order. Where SCAT, anything beyond 1 is nothing that should be practiced. The only practice thing is SCAT 1. And that is what it is. Even if you look at it, sir, in the pancreatic cancer, what I was talking to is, we have to, even the SCAT 1A, like let's say the BRCA is positive for pancreatic cancer, we have to look at the quality of data that is evolving from the trial that has proven this fact. And then we should look at it and then decide, rather than just that because a trial is proven, it's a prospective randomized trial, it automatically fits into 1A. So that also has to be correlated with the quantum of benefit and the toxicity it gives to the patient.
Okay, so this is another review on the SCAT scales themselves. This is again from the magazine of European Medical Oncology memo. So we'll look into, so some of the points what you raised and Dr. Raju said is coming into here. So I'm going to turn to Chirantan on this. Particularly you spoke about the applicability of an SCAT, in which you said a new point. I'm going to ask you some doubts. These are the review on the SCAT themselves. The score depends on the current level of evidence and thus can change over a period of time according to the newly available data. Number 2, in MTBs, SCAT can allow to identify off-label use may be justified in MTBs. That's a very important use. And then you have to, then what, Sahu and Aju said, they slightly have a difference of opinion. Individual patient setting, whether we can move on to SCAT2 for a given patient or not. Now, however, to date, there is no publicly available overview of SCAT score. That's the comment. So, Dr. Chirantan, and then there is some criticism on SCAT itself. I'm coming to you, Dr. Chirantan. You mentioned that it's you can go in and do the scoring in SCAT. I'm not very sure about that. One of the criticism of the paper is that the SCAT score is not publicly available for you to go in and work around, for example, like OncoKB. Can you clarify that point, Dr. Chirantan, and also address the previous memo points on SCAT?
Yeah, yeah, sir. Actually, no, I was referring to the freely available scorecards, the MCBS scorecards, not really the SCAT scores. Actually, if you see that, yes, yes. So actually, sir, MCBS data is very limited. There are very limited number of drugs, and also, as Dr. Randeep was saying, where we want to see the actionability, CDK4/6, PIK3CA for certain organs, like if you select PIK3CA, it will not give you anything other than breast. Okay, so how do you apply that in lung cancers? That is not possible in SCAT. But there is actually one data. So what we have done, we have applied a little bit more of back end work. If you see that paper, that SCAT guideline paper, it has got a table which has got various different, sort of public databases that you can excavate and you can mine the data. So we have tried to develop some API interface tools and develop our own software. So when we get a genomic alteration, we put it into that and it kind of mines through all that, because manually it's not possible. Then you will need at least one human resource to go into each of those. Somebody goes into CBio portal, somebody goes into clinicaltrials.gov, and so on and so forth.
Point taken.
So
I think one of the panelists already supported you, saying that, but I wanted you to clarify how workable is for an individual.
Sir, SCAT, as of now, if you give it to, without tech interface, it cannot be used in day-to-day practice.
Actually, the OncoKB was developed and floated by Dr. Chakrabarti, and she's one of the lead, lead evolutionist of the SCAT itself, and she sits in MSKCC. And whether the SCAT can be made available, but I'm going to turn to one important question. I think Dr. Kumar already mentioned that one key concern that everybody says is that it's not fast enough to account for the rapid changes and emerging molecular targets. It may lag behind. And then the next big question is that it talks about individual genomic alterations, but once you put it into the context of an extensive or a more elaborate molecular profiling, where you see multiple mutations, this seems to be completely not relevant. So I want to talk to Dr. Kumar on his viewpoints on, I'll come to Randeep also, on why SCAT is not able to address these questions. Why is it not fast enough? Dr. Kumar, please.
Sir, the moment we have a matrix, one of the inherent, for example, if you take up to date, whatever you get anywhere published, same night you will get it. Because what they simply put the evidence which is there. They don't get into the judgment part of it.
Right.
The moment you get into judgment part of it, that metrics you have to justify. And that process takes time. And that is where any metrics, when we apply, we need to be very careful. That doesn't mean I'm saying I'm against metrics. Metrics are useful, important. It gives you guidance. But then metrics should be nimble. And nimble enough. If not, the disclaimer should be there. And that is one of the problem with any metrics.
Right.
Maybe
I think you have said both. You said it has to go through a review process, but I think you use the word nimble. It's a challenge to be both nimble and fast and be detailed. So it's always a trouble. I think Randeep and Aju raised their hands on this particular issue. Then we'll have to quickly move on to the scales of MCBS. Do you have a point, Dr. Randeep?
Sir, so my point was what Sahu was trying to say. Even in this paper, if we see the SCAT scale of 1 has been given to around 68% of the trials. But when we
are going to come into this paper, we are going to come into.
Okay, so usually I want to say that they are correlated. For us as a clinician. I think ESMO MCBS is more beneficial rather than SCAT. SCAT just shows alteration. But how much data and in what setting and how many numbers of patients and what trial and what surrogate endpoints have been used, that is what MCBS partly takes care of.
So you have already answered the next question to me. I'm going to come to that. So now we are going to look at the MCPS. As Dr. Kumar mentioned, this is version 1.1, which is itself an improvement from version 1. Again, this was in 2017. In the year 2020, an MCPS has appeared for hematological malignancies also, as some of you may know. But this is purely for solid malignancies 1.1. The beauty is it's easily you can look into it, you can work around it. There is a bit of a charm to this, and it is divided. The whole evaluation is curative and non-curative, and you heard about the ABC and 1 2 3 4 5, and the curative intent and the non-curative intent. The form 1 talks about curative intent, and the non-curative intent where they look at the curative intent is on neoadjuvant and adjuvant therapies, and the non-curative intent, if you have a primary outcome based on OS, it's Form 2A, and if your primary outcome is based on PFS, it's Form 2B. And like Purvish sir mentioned, single arm trials also has got a Form 3, and now they have added one more form, that is for, this is the latest addition, quality of life checklist. These are the forms. You can go into these forms and you can play around with these things. So this is definitely a little bit more versatile. And so I want to ask Dr. Aju. So Dr. Randeep just mentioned that MCPS is more applicable clinically than SCAT score. On that point and on this, what do you think, Dr. Aju?
So I must say I have a conflict of interest here. I'm part of the ESMO MCBS extended working group member since 1 year. So I'm
very good for the disclaimer, but you
privy to the discussion. So I'm biased towards the ESMO MCBS in that it is actually a better reflection of how of the quality of a clinical trial. The SCAT is a symbol of is a representation of the strength of the evidence that is existing. Now, the ESMO MCBS penalizes or rewards trials based on the competence or the lack of bias or the kind of endpoints that are being used. So in that way, the ESMO MCBS is, I think, more robust. So like I always imagine, the specificity is good, which means if the ESMO MCBS scores high, then I feel the study is pretty good and the trial is good, it's well done. But just because the ESMO MCBS scores low, like has already been mentioned, for example, in single arm trials, doesn't mean that that drug or the target is useless. So that's my perspective.
Thank you. I think you are one of the, workers on this. You are one of the engineers who are constructing this, but very nice that you said that you touched upon. So let's quickly overview. This is the Form 1, and as was mentioned, there are some metrics on which improvement of in OS after 3 years and improvement in DFS if OFS data is not mature. And you can go into complete depth of each of these forms. It's all available. And so I'm not going to elaborate on this. Is Form 2A for non-curative intent, Form 2A, 2B, and 2C, and then they have these scorecards. But the beauty is this: you can go inside, and this is one example. So I want to look at what is amivantamab today in lung cancer in the palliative setting. So do I get some information out of this? So you can just key in these questions, and it comes out with it gives me a score of 3, and it says why based on these trials. Not only that, it clearly then captures at the score of 3. It is because it also tells me the PFS, and everything is almost ready-made, beautifully engineered, and given to you on a platter. So, Dr. Amit Verma, do you routinely use MCPS, and how relevant is that in clinical practice? Is it completely infallible? What do you think, Dr. Amit?
So I personally do not use MCBS on a routine clinical practice. That is my personal take. But this is a very fruitful tool which I can see right now with a lot of information on the platter. especially when you have to inform the patient in terms of the benefit and the cost involved of the drug actually. Especially the quantum of benefit can be easily documented in this case. But I have to tell you one thing very important. If you're talking about a single genetic alteration, fair enough. But the nuances of the alterations are not captured by any of these scores. So those things has to be a lot of disclaimer while we speak to the patient that these can change. Even if a drug is promising or a target is promising, an alteration in a different location may have a different outcome altogether. So all those things are not covered in these particular tools. But yes, it is a very handy tool for the patient to talk about the benefits and the limitations.
Thank you, Dr. Amit. You touched upon one of the most important points, why there are still something for that. And I'm coming to Dr. Kumar. There is also equally robust criticism on this ESMO MCBS score, even though everyone seemed to like it. And let's look at this paper by Dr. Gawali and biases of the ESMO MCBS. And of course, Aju is sitting here. But these are the important biases they have pointed out. There are more than 7 shortcomings in ESMO MCBS itself. Number one, evaluation of the control arm. If the control arm is inferior, you are going to get a very good outcome, and this goes for, I mean, any trial outcome, but this is particularly with regard to the scale. If the control arm is not good, and if in some trials crossover is not allowed, whereas crossover would have been the best option to do. And if, for example, if control arm is chosen as a placebo, which is not a standard of care, that may give a very good hazard ratio, etc., and they may graduate into a very high MCBS score. And then, what is the inferiority, non-inferiority margin they chose, and how do they look at the post hoc subgroup findings and censoring and all that one. And these are all the important criticism of ESMO MCBS, which may be true for interpretation of many of the trials. But then, Dr. Kumar, what are your comments on this?
So I agree with all the criticism which is there for NCBS, but that remains the same for any individual trial. And we talk about all these issues for any trial when we talk about just because we have a score, and sure the score will have those advantages and disadvantages. So these are important ones, but it's a good beginning. But it needs a whole lot of changes, and as you heard from everybody, by and large, no one uses in the clinic. This is a bit like IDSA, a guideline for febrile neutropenia, when you want to score it and put in low risk group and high risk group. But if you ask any clinician, do you put a score and then put a patient in high risk and low risk group for febrile neutropenia? And I tried doing it. That was around 15 years back, and I didn't hear any clinician who was doing it. So it is good to have it is a good beginning. We have a score which helps us in understanding things. But to have being clinical relevant, it will require certain refinement to make it easier for it to interpret, and also legible. Not legible is the right word. Where we know that, for example, I give you, wherever the ESMO, MCBS puts the score for anti-RK inhibitor or RET inhibitor or ROS inhibitor,
Absolutely.
doesn't matter.
Yeah, we are going to come to that, Dr. Kumar.
Yeah.
So why I went into the elaborate criticism of both, because this is, now we are going to look at the paper, and this paper has taken SCAT and then ESMO. That's why I wanted to really, look at the, what do you call that, weaknesses of these two metrics itself before we go into the paper. And now we will look at this JAMA paper, this is the paper.
Sir, just a quick
Yes.
one liner.
Yes,
The
sir.
only thing is that these scores are looking into actually what has been approved, and they are giving a score on the thing. So I think what
I agree. That's how where I'm taking is that,
yeah,
based on these two scores, this paper has been developed, and then we are trying to look
No,
at
no, no, no. I was just coming to the criticism by Dr. Gwali. That's fair enough, but these score are meant to look into the level of benefit for patient care on the approved drug. And what the criticism has been there is basically trial specific, which is actually the job of the FDA or the regulatory authority, and they should actually not let these substandard trial approvals go through. And if that doesn't go through, these scores will not be there in the ESMO or the ASCO, whatever is there. That's the small point which I want.
No, to counter your point, Sahu, what
Yeah.
happens is that once the trial is published, for example, there was a discussion on the ESMO MCBS, and I mean, from which
Yeah.
this particular paper. They say that suppose a trial has been performed with a placebo. Let's take that example, and then you have a very high, difference, delta, and then there is a very good hazard ratio, and that it gets published and it gets approved. Now
Yes, sir. So the approval
doesn't go into the detail of the trial, but
sir, but the approval. But if the trial is weakly done, it's the job of the regulatory authorities not to get the approval. That's the point.
Point taken. But once that mark is taken, even if you score mark in an ordinary college, and if you still show 99 mark, and that mark is taken for your selection, that's the point I'm trying to make. Your point is also taken. And now these are all what Dr. Purvish sir showed in the JAMA paper and in detail. So now, as he pointed out, what is, this one is that what is this JAMA paper is trying to tell us is about what are all the, indication it looked at. He went into his detail. But the most important point that we have to, before we consider whether this is going to be a practice changing paper, or that 38% of the approval, accelerated approvals, and 62% were regular approvals. Of the 52 regular approvals, 25% of the approval were based from single arm trials, based on which these approvals. And so what is the implication of genome targeted drugs getting approval on early phase or single arm trial? So first we need to address that question. Dr. Sahu, can you answer that question?
Sir, can you come again, sir, please?
See, what is the implication of many genome targeted drug approvals on early phase or single arm trials?
Sir, I feel it is okay. I don't have a problem with that. Like Dr. Kumar told, the craniopharyngioma thing, you have to look into the disease specific and what is being shown and how common it is. But I have a problem with trials being approved on response rates alone. And Randeep spoke one of those things. And that is the trial. And if you look at the paper, more than 50% of these agents have been approved on only response rates. And the cutoff of response rate sometimes is too shabby. A 20% response rate or 15% or 22% in a second, third line could be a continued effect of the on the previous therapy also. You never know in a single arm trial.
So the trials that which this JAMA paper looked at, as you pointed out, 55% of the approvals were based on ORR, and then only 8% of the approvals were based on the OS improvement. That's the kind of trial this paper itself has looked at. You pointed that out. And now coming into the analysis of this JAMA paper, it has distributed the trials on this SCAT score, and then it has distributed the trials on the MCBS. What it has done is it has overlaid the MCBS score on the SCAT score. That's the method it wanted to say, and based on that, it says that only 29% of the genome targeted drugs meets the ESMO MCBS threshold for a substantial clinical benefit. That's the final interpretation. But how did the doctor arrived at this? By overlapping SCAT and ESMO. So is that a valid way of doing things? That's what I am worried. That's the question I wanted to first ask Dr. Kumar. You have one metric and it has its own deficiency, what we saw. You have another metric that has its own problems, and then you overlap both and you try to derive at another final outcome of saying that only 29% are clinically useful. So is that methodology agreeable, Dr. Kumar?
Sir, I agree with you what you're trying to say, that one metric was for evaluating one aspect, that you have mutations well defined and you have a therapy which is available in that setting, it works or not, and that's what it defines. Second one provides the quantum of benefit. And if you take the first part, it looks like till that time, if you take, I tried to get into all the trials, but those details were not there in supplementary. I know I was very keen on knowing which trials they were saying, and then we could have had a good discussion which kind. And I'll give you some examples, and so that can I know it looks like most of us are talking the same thing, but having said that, second part was the quantum of benefit, and that is where we have an issue regarding how we are defining to give a hint to our clinicians and maybe other people who decides treatment being available for our patients, and where it has a potential to go wrong both ways. Now, for example, the areas where I work in, I'll keep you giving you example one by one. Salivary gland tumor, HER2 positive, TDM1, 90% response rate, 10 patients data, it is part of most of the guideline. Next one, you want thyroid cancer, anaplastic CA thyroid patient, BRAF mutation positive, single arm study, around 60-65% response rate. No other treatment available. RET mutation in papillary CA thyroid, RET mutation in medullary CA thyroid. There are different type of mutation. Response rate similar, around 70%. Again, they are part of a routine guideline. If you take another one, I told example of aminoblastoma. I gave example of papillary craniopharyngioma. I'll give another example, uveal melanoma. There is a unique drug called Tibenta First, which is available for HLA. I'm forgetting the what they call subsequently.
I think you have made the point, Dr. Kumar.
So idea is that.
Correct.
Yeah.
I'm
so
also
idea is that. Can I make a last statement, sir?
Yeah, please.
So idea is that is where the question comes, and I'm sure the way NCB, they changed after having a modification with all this more information, they will change again. Because I'll say anyone in lung cancer, will they prefer anti-RK inhibitor or docetaxel? That will give you a hint.
Absolutely, that's a way. I'm going to come into that a little later. That's why I'm sorry about that. But you have made a very important point. We are going to touch upon those deficiencies which are not addressed by. This is a very important criticism published by Peter Horak on this JAMA paper itself. And this is the conclusion. And he himself is a translational oncologist. Flawed conclusion of the JAMA paper. That's what he says in one line. Why? Because SCAT is a different scale and MCBS is a different scale. They were independently developed for two different applications, and you put both these things together, and then you try to arrive at your own magic number of 29. That's exactly and what Dr. Kumar said. That's exactly the point Dr. Horak makes. Look at the example of larotrectinib and entrectinib, and then what you mentioned, RET. So and then MCBS gives a score of 3, and so many examples that you pointed out. These are all not essentially well captured into the MCBS scale. So, and we know very well that many of the, our patients, they have very unique mutations, and therefore they can only be looking at a possibility of a small patient cohort trials wherein these scales are not able to answer. That's the criticism. So, Dr. Randeep, what do you think? One word about what is the criticism? What is your comment on this criticism on the paper, and what is your take on the paper, Dr. Randeep?
Sir, I actually echo what Kumar sir just said. It all depends upon the scenario, and that's why I also request Purvish sir has sent the supplemental material. I did go through it, could not find what conclusion has been based on what trials. I don't think so I will change any practice, first of all, based on this. But I also feel that both the scales are independent. We can apply both SCAT and ESMO MCBS, but over and above, what is more important in today's era is that you can't just go through the scale and do it. You have to go through the paper. You have to, suppose tomorrow I'm using anti-RK, it is always better. If I'm using, for example, TDX for any tumor like esophagus or any rare tumor like anal cancer, HER2 positive, I also need to go to see that paper, that in that basket trial, how many anal cancers are there. It is always a good idea. We cannot just go SCAT MCBS and just apply to the patient. It is always.
I'm going to come to you on that particular trial. I'll come to you, Randeep. And allow me to interject, sorry about that. And I'm going to look at a little bit into Dr. Taibo, and a great career, completely made out of, looking into the clinical trials. She has many publications, and JAMA 2018, 2016, everything is about, trying to interpret the trials based on these kind of metrics. That's what she has done. And the clinical benefits of not only targeted drugs, but cancer drugs in general. Just want to tell everyone that most of her publications on all the approved cancer drugs, you know how much mark she would finally give? Is only 20 to 30%. None of them could get a pass mark. If you consider 40 as a pass mark or 50 as a pass mark, all the publications. And then she has also looked at a lot of authors' conflicts of interest and financial conflicts. So very strong career. And she was formerly in Spain. Now she has moved over to Harvard, and she is heading the Boston portal, which is a very important regulatory authority, and she is a great career. But that is the publication record of Dr. And for example, what she has done is she has overlapped NCCN guidelines and ESMO scale and arrived at a score, which is again, that's the methodology she has used in everything. So my question is different. I'm going to come to Dr. Sahu. Why can't we do these? She takes these papers and puts these metrics and comes out with great publications. Dr. Sahu, why can't we do these things?
So we can, I think it is as Dr. Kumar will agree, partially may not, it is the job of the academia in the country to do this, because they have the privilege and the facilities probably to do that. But in India, we don't have a culture of...
No. This is very easy. She just has a computer and she has to...
Sir, I was coming to that, sir. So in India, even the academia is somehow unfortunate that they have to devote 80 to 90% of the time to patient care. And these doctors do not have to. They devote 50% or more to clinical, developing clinical regimens and trials. So I think our culture has to change. So that is what
Totally. Thank you. I'm going to come into clinical cases, our own experience, and then we take it into this. All of us have seen this. We have had great cases. I'm only talking about genomic targeted therapies, and we have also had not so great cases. So let's look at this paper's interpretation based on our own experience. There are excellent examples, like what I'm trying to show: osimertinib, first line response; dabrafenib, trametinib in CA colon in BRAF, and entrectinib response, like what Dr. Randeep said and what Dr. Kumar said. This is pembrolizumab response in CA ovary with PDL2 in the NGS, and pralsetinib response and selpercatinib. So these are all great responses that tells us that once you get everything right, definitely this is the best way to treat and all that one. But then comes the difficulty, not so good cases, difficult cases. That's when you start wondering why it's not always possible to be successful with targeted therapies. Let's go into that. So I'm going to give some examples. This is a patient, lung adenocarcinoma, has got a classical EGFR mutation here, exon 19 deletion, 75% VAF, but has got an amplification of ERBB2 with CNV of 7. So Dr. Amit, I want to ask you, this patient was started on treatment with osimertinib, pemetrexed, and carboplatin, and did not show a response. So
did not show a response, or did show a response?
Did not show a response. That's
okay,
why. So we are all always say that target. We spoke great about as being, but then we are completely stumped. That's when these papers become relevant. So I'm quoting an example. So it did not show a response. Look at it, it's really slapping on the face. So what do I do?
Well, we have two targets here, and we chose the EGFR in this case, and ERBB2 was ignored actually. So there are implications of the second alterations, which is causing resistance to this particular scenario. But none of the ASCAT or the MCBS will guide us to target two
Exactly.
genomic alterations.
Exactly. Nobody is going to guide us from, into this. So another patient, this is a very single target. This is an IDH1, very classical mutation in cholangiocarcinoma. But, ivosidenib has not touched this patient in second line. And this is another patient. This is a patient, again, a CA lung with an EGFR L858R and a T790 mutation. At the same time, you can see, tier 1 mutations as per the SCATs, but has no response to osimertinib. And, I don't know whether the AR amplification has got something to do with this or not. So one more patient. This is a patient, this is a lady who is a 60-year-old lady who had a CA lung. And she has an EGFR, L858R. And this was diagnosed in, January, February in 2022. And this patient was appropriately started elsewhere with osimertinib, did not respond, and then moved on with PEM carbo, short PFS, and then comes to us in October 2022. We suspect something is going wrong. We do a repeat NGS, and this is what we get in the liquid biopsy. And so what do I do with this, Dr. Chirantan? And it's still showing in EGFR 8L858R. And this is the liquid biopsy. And I've given you the previous thing. The patient is not touched by osimertinib or not great with PEM carbo. What do I do with this? And what's going wrong here?
Sir, first of all, just want to clarify, this is a liquid biopsy report?
This is a liquid
that you're
biopsy. The previous one is a solid biopsy.
Yes. So actually, sir, I mean, there is, of course, lot of variations in the coverage and in the detection of mutations between liquid and tissue-based. We know that. I mean, I would have, it seems this is a foundation report, and I would have loved to see that if the foundation report would have been a tissue-based report, because obviously there is some molecular mechanism underneath which is not allowing a EGFR TKI to work and is also giving a suboptimal response to the chemotherapy. So that needs to be detected. But of course, the problem of tissue being an issue is significant. Maybe there is somewhere a PIK3CA alteration sitting somewhere deep underneath, which is...
Yeah, the point is that, so if you read an NGS and it's not telling us everything, and that's what you are mentioning, and this was just before you joined Foundation 1, before that, so the point what you have made is that the report is not telling us everything. Something else is going on in the tumor. And actually, this patient had an excellent response to amivantamab, and the patient is now on amivantamab for more than 1.5 years. That's a different thing. And why I'm quoting this, another patient, this is a patient with a CA lung with ERBB2 HER2 amplification. And so I'm going to ask this to Amit. Amit,
Sir, one point if I can make, just to add, sorry, is that the actionable mutations and digging into the data of actionable alterations vis-a-vis resistance creating alterations is a very important factor.
No, we are coming to that. Why all these things is where? I'm just quoting some random examples of where...
Absolutely. Your first case and this also shows ERBB2 amplification is a well-established resistance mechanism to anti-EGFR, both in therapy naive as well as an acquired alteration. So that's the only...
Again, this case is another one that stumps. Whatever you mentioned, ERBB2 is this is another amplification, copy number 6. And this patient responds greatly to afatinib for more than 3 years, just with afatinib, which is completely contrary to the previous patient, and has progressive disease. And now we have put the patient on NR2. NR2 is not touching the patient. So the point that I'm trying to come is this, why there are so many disappointments, which is what I want to ask. Amit Verma, can you answer that question? That
So I would
one
like
claim.
to know. I would like to know what is the current genomic alteration post progression of afatinib actually? Has it changed?
No, I think we don't have that. That could be
So
the
afatinib, yeah, if
could be the
afatinib
case
has
you are
worked
going.
for ERBB2 and it did good for the patient, that's great. But something else is also developed on the go, which we need to identify and can explain why Enhertu did not work. So maybe that it is no more ERBB dependent cancer further, and it can be just a wild type or some significant alterations or different clones altogether as of now.
Absolutely. So reading the NGS and that being deficient is what you and Chirantan pointed out. So therefore, I'm trying to tell that our reading itself is imperfect. So why blame finally? Why blame the drug? Is where I'm coming at. So Dr. Aju, I want to ask you, how do we find solution with this so many imperfections? The trials are not good, scales are not good, recommendations are imperfect. How do I face this in the clinic? What do I do to overcome this when I want to do genome-directed therapy? What are the ideas? Dr. Raju, can you tell us?
Yeah, so what I can gather from the discussion and understanding the literature as best as I can is number one, SCAT is just a guideline or a guidance for a practicing clinician into the strength of evidence. ESMO MCBS is more or less like a student appearing for NEET entrance exam. You have to refer to the previous question paper to understand what works. So it's ESMO MCBS is sort of a guidance for pharma companies in terms of how to design better trials that are patient-centric. So if you are actually, if a company wants to meet a high threshold, then you have to meet certain thresholds. So, so that is why the ESMO MCBS is trying to address these shortcomings, the biases, etc. So it is not relevant for a clinician practicing in the clinic, but probably will become relevant for the bigger system itself. Number three, precision medicine actually requires an understanding of the target, finding the right drug, and a group of people sitting together, talking to each other, because none of us are experts in anything, in everything. So a molecular tumor board to figure out what to target and how. And finally, we must stay humble because we don't know anything about anything yet.
Absolutely. I think your paper is great where you have told, I'm always fascinated by your paper. Wherever I go, I put your paper first to see, what is the final use in at least in Indian context about all these things. But then to Dr. Randeep, whether agnostic approaches again don't seem to be helping. That's what you also pointed out. Agnostic approaches, just because there is a target doesn't mean anything. In individual tumors, they seem to be responding differently, and there is same target. We know that now agnostic approvals have come, but then for some funny reason it works beautifully in some malignancies, let's say in salivary carcinoma, but it works poorly in some other malignancies. So, and I think this is what you wanted to touch upon, Randeep. You said that NR2 again does very well in some places in the same setting, second line or whatever, but does very poorly in some other setting. So where is this whole concept of agnostic approval itself? Even this NR2 is too good in some place, too bad in some place. So how do we interpret this, Dr. Randeep?
So to my mind, it is very difficult. I mean, if we have to do it, I mean, I understand we have to bring I mean, for the regulators and for the researchers, it is very important to bring the drug in time rather than, I mean, bringing, for example, we are bringing HER2 positive. We were never aware that HER2 has a significance in endometrial as well as cervix and ovarian cancer, for example. And tomorrow I have a patient who is now on palliative care and HER2 positive ovarian or maybe endometrium. I am trying a TDX and I'm doing that. So that is one point of looking at it, that we bring the research the research to the patient benefit. But on the other hand, the trials shall continue in the background. As you said, the approvals, the regulatory full approval has to be very cautious. It can be an accelerated approval or a breakthrough designation. But then if today I know that HER2 positive endometrium has a role, then we shall also have a bigger trial Endometrium is not something which you can't get HER2 positive endometrium. You can easily get 100 patients by just combining 4 or 5 centers. So the only way is that once we have a breakthrough from the basket trial, confirmatory trial for full regulatory approval by including more number of patients shall be done. We shall not restrict there. And that is what FDA and other authorities shall also do.
The other trouble with NGS interpretation and acting on the genomic level is that, as Dr. Amit pointed out, presence of co-mutations, definitely we all learned that it can have negative effects, and then emergence of resistance. Dr. Sahu, do you think that emergence of resistance and co-mutation is why sometimes we get, or many times we get disappointed with targeted therapy? What is your comment, Dr. Sahu? Dr. Sahu?
I think Sahu had to leave.
I'm sorry about that. Dr. Amit, can you say that inherently as we have evolved over billions of years, right from a single RNA molecule, and we have gone into mutation after mutation to finally evolve into a human being over the past 4 billion years. And the central theme is that if you look at how human beings themselves came, each time an extinction level event has led to mutations to finally. So this RNA has been going on for 4 billion years. So it's very naive on us to expect that it won't retaliate, the tumor won't retaliate. So that's why we are failing in many ways. What do you say, Dr. Amit?
Yeah, so I have to add one more aspect to the whole discussion, that whenever we talk about genomics, we tend to lose some of the information. To give you an example, like bladder cancer, which is to have but fail to identify the pattern of positivity makes a significant difference in terms of the outcome. So whenever you do a genomic study, all the heterogeneity is being combined into a single test and the special expression is missed. So I think that may be the reason why you fail in certain scenarios and it works in certain scenarios. So having said that, definitely protein level information, RNA level information, DNA level information, all is required to be certain that yes, it is going to work. So this is my take home. And further, coexisting mutations do make a difference. But as we have very strong drugs available like NHER2, the coexisting mutations are being overcome as of now. Like if you have a ERBB2 with PIK3CA, the standard Herceptin or TDM1 may not work, but NHER2 may work in these cases because the conjugate is too strong. So we have to learn the capabilities of the efficacy of the drug and apply it accordingly. So again, some nuances for the tumor board to decide on.
Okay. Amit, Purvish sir, can I take 5 more minutes? I'm at my time exactly 5 more.
Yes, please go ahead. Please go ahead.
Amit, like what you pointed out, why do we don't do well? And one of the example is it's combination therapies. I want you to take this example and the case that I'm going to show you whether if you address multiple targets, whether it's going to be successful. There is one case I wanted you to tell us. This patient is a urothelial malignancy.
So I can just see.
Okay. Are you able to see? Amit?
Yes, your voice is a little breaking up. Yes, urothelial malignancy. Yes, come again.
This is a urothelial malignancy diagnosed in April 2021. And I have given the amplifications and changes on the right. And then went on to have multiple, treatment intervention. It's an adenocarcinoma urothelial. And then by August 2022, developed these changes. And I'm just capturing the most important one, a TMB of 19 mutations. And we have NF1 and RNF43, urothelial adenocarcinoma, where I have already crossed through, FOLFOX-like regimen, GEM, and gemcitabine-based regimen, FOLFOX-based regimen, and even nab-paclitaxels and all. So at this point, I have this, if I have to think about multiple targets, what will you do, Dr. Amit?
So this is a very interesting case with high TMB and already have IO has been attempted and failed actually. And the explanation can be seen through the STK11 mutation, what I can see upfront, that can explain why it did not work. Having said that, the NF1 duplication, what we are seeing here, may suggest to me that this may have HRD to be higher actually, HRD score to be higher. So I would like to know what were the responses with the platinums, what you tried earlier. Was it substitutable or not?
Everything was few months PFS. Everyone went on few months PFS.
Few months. So in this case, STK again is known to cause chemo resistance, which is also again evident and explained by the clinical course. So targeting STK is something what should be tried along with the chemotherapy if possible. So we can use mTOR inhibitors, and there are certain trials where we have STK11 mutation inhibitors available these days in the US under trial setting. So I have used one of my patients to go over there for the head and neck cancer. But having said that, yes, STK is a very important culprit for the resistance to chemotherapy and IO together. And it can be easily overcome by the easy drug available, that is everolimus, what we have. But monotherapy everolimus will not work in this case. You better try chemo with everolimus or IO with everolimus in this case. There are certain...
Thank you, Dr. Amit. Similarly, and quickly taking over, NF1 mutation, we looked at NF1 mutation, and what we did, after elaborate, like what you pointed out, we combined pembrolizumab with trametinib, and then we had a 1 year plus PFS on that. The point that I'm trying to make is that, like what you said, every tumor is unique, and therefore all these previous trials, I think that's what you illustrated. That's what I want to bring, not to show about that particular case. The point that I'm trying to make is that every tumor is unique, so we need to do more. We need to do genomics. We need to do a completely different profile of clinical trials. It could be basket trial, and what Chirantan pointed out, basket in basket trials, and the Tapur design, and now we have this I predict and match. So unless we match multiple targets, we are not going to get to the desired outcome. Is the broader message. That's what I wanted to come to. So I've completed my questions, but I'm going to come to my panelists. Finally, Good, Bad, and Ugly is a Hollywood movie. But in India, when we do Bollywood movies, even if the actor has to do multiple roles, finally all of them will be good people. You will never have a bad, our heroes can never be bad people. That's what we do. So in that context, genomics, is it good, bad, or ugly, or is it all good? Dr. Sahu, what is your one-line answer to that? And I'm going to run through my panelists. Is genomics really there to stay, go good? And are we still deficient in understanding that? What is your take, Dr. Sahu? It's your opinion.
Sahu is not here.
Dr. Randeep?
Sir, I will say good, but if used wisely and in appropriate hands.
Dr. Raju?
Unlike what the author thinks, I feel genomics is good as a practicing clinician. As somebody sitting in a chair, evaluating studies, I think it's easy to blame it all. But I think genomics is good for the patient. Yes, no doubt about it.
Dr. Amit, genomics is good?
I think I strongly believe in genomics, so it is very good. It is just that we are learning how to use it better.
Chirantan?
Sir, why ask me? It is my bread and butter, so it has to be good.
So the final authority is Dr. Kumar. So that's why I asked the last question to him. Dr. Kumar, are we doing it right? Are we in the right direction? So what is your final word? Because, without genomics, I don't think there is any more oncology. That's what I think. But what is that you think? You are a wise person. You have so much experience. You have seen, success, and you have seen in the good and bad of it. What is your verdict, Dr. Kumar?
Sir, thank you for kind words. So, sir, it is good as we heard from majority of the people, but we have understood very little as on date. And that was very nicely said by someone. We need to be humble. That we should understand the limitation of whatever we have understood till now. I'll take half a minute to give example, and I give example again from head and neck cancer. I gave all the examples which I said it is good, and I am sure everyone will agree. And where it is not so good or bad is same head and neck cancer as squamous cell carcinoma. I have done fair number of sequencing yet to get something where I can say I am comfortable having a targeted therapy based on genomics. There are targeted therapy, I concede. So yes, we have learned little, and some of them are really good, which we all use it. We need to be careful about it, judicious about it when we use.
Those are really the wise words. And, my panelists, thank you, all of you. If I have, sometimes interjected you just for the flow of the time. Eminent panelists and interviewees, and it's my very humble honor that I have been put to ask questions to you. Amish, thank you so much for this opportunity. Back to you.
Thank you, sir. And with Parikh sir's permission, I think we are at 8:40 right now. So I'll just take 2-3 minutes. But before that, congratulations to Raja sir. Whenever I hear you moderate and present your slides, only thing awesome is the word which I always say. And I don't think, this is somewhere even closer I'll ever come to that unless until I take tutorials from you. Today, interviewee, and entire panel discussion, in fact, made me proud that we have created OBTO. And if you see each and every chapter of OBTO, actually we start from SCAT, then we go to MCBS, and then finally, as Tarini says, we try to dissect out how much is the magnitude and what is the applicability in the clinical trial. And that is what exactly each chapter of OBTO does. And I'm so proud that we created that. About today's discussion, I feel SCAT gives us the quality threshold and the richness of data. And it also, I think, gives us medical legal cushion, especially in an off-label drug whenever we want to use it. Of course, SCAT lags behind the evolving data. As we have more and more data, like the NCCN, as Kumar said, immediately the next day you have the next version. SCAT does not have that cushioning. MCBS, on the other side, I personally believe that for the very beginners and people who do not want to read, or those who has less time to read, for them MCBS reduces the bias and it gives the accountability for reasonableness. Yes, of course, there are many shortcomings, and there are other associations like Association for Molecular Pathology also. And we have now the NGS reports coming, likely pathogenic, or less likely to be pathogenic, or VUS. These are the things which AMP qualifies. So similarly, in future, I hope that we will have an OBTO scale also like that, or the score also like that. But my final take on today's discussion is that there is no doubt, we right now do not possess enough knowledge about molecular pathways in cancer. Let us be humble, because we do not have that knowledge. But till the time we have that knowledge, SCATs and MCBS is a good guide or the good tool to go forward. Yes, it is not sacrosanct at all. We need to have more discussion, especially as Raja sir showed about the resistance mutation studies. There is co-mutations and resistant mutations, whether they are bystanders or they are causing resistance. We still don't know. And we need to have more molecular oncologists in our team to understand that. And with that, I close my final review of this, and over to Parikh sir.
Thank you very much, Amish. Thank you very much, Dr. Raja, Chirantan, Kumar, Prabhash, Aju, Amit Verma, Randeep, and Sahu has already left. I just want to end by reminding you that our next OBTO will be on Tuesday, 12th of November at 7 p.m. and hope to see all of you again. Good night. Have a wonderful evening and dinner.